All right, everyone. Welcome back to Bio 1120. Okay. Hey, remember on Tuesday, I started talking with you about metabolism, how it is that organisms have a certain set of chemical reactions that take place within them, and how it is that they cause certain favored reactions to take place more readily than the ones that they're not that interested in. So today, what I want to do is, now that I've talked with you about the thermodynamics of it to a large extent, let's get to work and talk a little bit about what it is that the actual, how it is cells are causing some chemical reactions to be more likely to occur than others. That's the agenda for today. And so what I've got for you here for the lecture outline, it's the same one that we talked about on Tuesday. And on Tuesday, I ran through pretty much to this part of the lecture outline, but I didn't tell you all that I wanted to say about ATP, so we'll start more or less with that. But remember this slide, right? This one here is showing you again, just in broad brush strokes, what's going on with a reaction that is energetically favorable, okay? Bottom line is, if the delta G, the change in Gibbs free energy, is negative, what's going to happen is the reaction can occur spontaneously. It's something that will allow you to move quickly from substrates or reactants to products, and along the way releasing energy. That's going to be true for all spontaneous reactions. And the funny thing is a chemical reaction cannot take place unless it is spontaneous. Right? So for a chemical reaction to take place, because of that first and second law of thermodynamics, we always have to go from more energy to less energy as the process plays out. And that's in contrast to a chemical reaction like this one. This chemical reaction is not energetically favorable. We're starting out with substrates or reactants that have low energy, and we're ending up with products at the end of this chemical reaction that have more energy, right? That can't happen. Nature, because of the first and second law of thermodynamics, isn't going to allow that sort of thing to play out unless you manage to somehow link these kinds of unfavorable chemical reactions to chemical reactions that can occur spontaneously. So when you put the two of them together, you may find yourself in a circumstance where things that shouldn't be able to happen do happen, just because we've married an energetically favorable reaction to one that was not energetically favorable. All right, so that's the highlights, I would say, of Tuesday's lecture. Let me remind you about some of the details for the energy currency inside of cells. Remember, glucose, that's the big chunk of change of energy for cells, 686 kcals per mole. Okay, let me let that sink in for a little bit. You guys know what a calorie is. It's the amount of energy that you need to increase the temperature of 1 gram or 1 milliliter, same thing, of water 1 degree centigrade. All right, think about that. 686,000 calories per mole of energy in a glucose molecule. Okay, a glucose molecule, right? 686,000. You can increase the temperature of a milliliter of water 686,000 degrees with the energy in 1 mole of glucose. Mind-boggling, right? I mean, are you thinking that? No, that can't be right. That's how much energy is present inside of a glucose molecule, an enormous amount of energy. Again, I likened it on Tuesday to more or less the $100 bill of the energy currency inside of cells. That's a big chunk of change. Cells are really happy to have something with that much energy and to store it and to preserve it and cherish it and show it off to their friends. Look, I got some glucose and you don't. But when you want to get stuff done inside of a cell, that's actually too big of a chunk of change. Just like a $100 bill doesn't work well in a vending machine down the hall, glucose has too much energy in it to be practically useful for cells. And what that cells turn to instead then is using ATP is sort of the dollar bill of their energy currency, because ATP, I'll show you the reaction in just a minute. ATP, when you break off one of the phosphate groups associated with ATP, you release 7.3 kcals per mole of energy. So instead of that really big 686 kcal per mole chunk of change, chunk of energy, ATP lets cells manage more useful units of energy, 7.3 kcals per mole. And don't forget, the whole purpose here is to be able to link that energetically favorable reaction, releasing a phosphate group from ATP with its 7.3 kcals per mole of energy, in linking that to an energetically less favorable reaction to have the less favorable thing take place. That's what this is all about. I hope I left you with an indelible memory about efficiency, right? When we talked about energy transfers, that second law of thermodynamics demands that every time you move energy from one place to another, from one covalent bond to another covalent bond, for instance, that you pay that entropy tax. That's what the second law of thermodynamics is all about. Internal combustion engines are hideously inefficient. That entropy tax that we pay when we turn on our car engines, 75%. 75% of the energy gets lost through an increase in entropy, the temperature of our car's engine primarily, as opposed to 25% being used for actual work, moving our car from one place to another. The transfer of energy from glucose to make ATP molecules is the single most efficient transfer of energy that we have discovered, period, right? Not just in biology, it's 34% efficient, okay? Over the course of a couple billion years, living things have worked out a way, brokered this deal, keep going to the bank of Mother Nature and saying, can you cut me a little bit of a better deal than you did yesterday, right? How can we work this out better? Billions of years of negotiating, 34% efficient process to convert the energy in glucose to more useful chunks of energy in ATP. But we need to do that again because the ATP is a more manageable chunk of energy and something that we can use to link up to other chemical reactions. So, for the first time, let me introduce you to a molecule that we're going to talk about a lot. This is ATP. That's the structure of this dollar bill of the energy currency for cells. ATP. A is the same A that we've talked about before. Remember how hip, cool molecular biologists don't bother saying adenine? Just takes too long. I don't have time to say adenine all day long. Instead, you just say A. It's the same adenine. It's the same A. We last talked about A in the context of being one of the 4 nitrogenous bases that you use to make DNA, right? So GATC, those letters that you string out to make a long chromosome that spells out all of your genetic information, same A, right? It's the very same molecule. But it's doing something a little bit different here, right? So you've got the nitrogenous base adenine attached to a 5-carbon sugar, the same ribose sugar that you use in the phosphodiester backbone to make a DNA molecule, right? Nothing different there. And attached to that 5-carbon ribose sugar are 3 phosphate groups, hence ATP. A, adenine, T, tri, P, phosphate, right? So this is adenine with 3 phosphate groups attached to it. That's what it looks like. An adenine with a sugar with 3 phosphate groups attached to it. Now, what do you remember about phosphate groups? Aside from the fact that I asked you a sneaky question about it on the last test. Was that number 32? That's the one that I still have pangs of conscience about afflicting you with that. What do you remember about phosphate groups? Serious negative charge. That's the thing I hope that for the rest of your lives, if somebody says phosphate to you, I hope that somewhere in the back of your head is my voice saying negative charge, right? Sorry, it has to be my voice. I wish I could have contracted to have somebody with a better voice say it, but negative charge. That's what phosphate groups are all about. Serious, intense negative charge. And remember, nature doesn't like to have things with the same charge right next to each other. Opposites attract. We learned that back when we start dating, right? Opposites are where all the action is. You don't try to get two negatively charged things together. Phosphate groups really like to hang out with positively charged things. And yet, in ATP, not only do we have one thing with that intense negative charge, but we hook it up to another thing with a negative charge, and we hook that up to another thing with a negative charge. You got three negatively charged things all right in a row. And so to pull that off, you can imagine taking two magnets with the same poles. It's hard to push them together, right? Unless you change the orientation of one and then they snap right together. But if you're trying to push these phosphate groups together to get them to stick, you need to make a covalent bond that has some energy associated with it. Guess how much energy? 7.3 kcals per mole is the kind of energy. That's, again, what's going to happen. You will release 7.3 kcals per mole for every phosphate group that you take off of an ATP molecule. So we have A, T, P, right? If you let one of those phosphate groups go away, right? You let one of the phosphate groups go away, doing that releases 7.3 kcals per mole. Good news, right? That's something maybe we can do with that energy. And in the process, what you do is you go from having ATP to ADP, adenosine diphosphate, and an inorganic phosphate molecule, just floating off by itself. You do not have to stop there. If you're really pressed for energy and you're a cell, and you've used up all of your ATP and you've made a bunch of ADP, right? You know what you can do? You can milk that molecule for more energy still. You can get another 7.3 kcals per mole by converting your ADP to AMP, adenosine monophosphate. Right? So you can push this a little bit further, but cells are in pretty tough shape. They've come upon really hard times if they need to be doing that conversion. Typically, you go from ATP to ADP, and it's only when you're really stressed that you'll take the extra energy from ADP down to AMP. Big picture, the most important point here is that releasing that phosphate group releases a manageable, useful chunk of energy, 7.3 kcals per mole. So that's what I've just been gesticulating about. This slide's showing you the actual conversion. Once again, you start out with ATP, you hydrolyze it, you bring in a water molecule, release an inorganic phosphate, you're left with ADP, and you get that little pulse of energy that comes out. The game now is going to be, what can we do with that energy to help cells accomplish things that they might not have been able to do otherwise? Because, I feel like a broken record here, because you can't have a chemical reaction take place. It will not take place unless it has a favorable delta G. So is there a way that we can hook up the favorableness, if that's a word, of this chemical reaction to a less preferred chemical reaction to cause the less preferred reaction to take place? Well, here is a specific example of that. You might recall from a couple weeks ago when we were talking about amino acids, I mentioned glutamine. That's one of the 20 amino acids. Did you ever wonder where glutamine comes from? I can't imagine that you did, but if you had, the answer is you make glutamine by taking another compound, glutamic acid, and smushing it together with ammonia. That's how you make that amino acid. That's the last step in making glutamine. But I got bad news for you, right? Don't pay attention to what's going on down here just yet. I know you guys are on top of this and you're probably already through with this slide, but focus on this. What do you see here for the delta G of this chemical reaction? To make glutamine, that process has a delta G of positive 3.4 kcals per mole. All you need to see right there is that it's positive. If it's not a negative delta G, it's not a spontaneous reaction. That chemical reaction can't happen. It's energetically unfavorable. If you put those two compounds, glutamic acid and ammonia, together in a test tube and you shook them up, you'll be shaking them up for the rest of your life and you won't make any glutamine in that test tube because the energy associated with that reaction is not favorable. All right? But you need glutamine. Right? That's one of your 20 amino acids. If you don't have glutamine, you can't make your proteins. You can't make your proteins, it's over. There's a clinical term for people who have that condition, unable to make glutamine. We call them dead. Right? So you need to have this chemical reaction take place. You're alive, you're here today, right? So somehow or other it's been played out. Here's how it happens. Here's how it is that we get past the fact that that chemical reaction has an unfavorable change in free energy. You know the answer, right? If you've been tracking with me for the last 5 minutes, the answer is pretty obvious at this point, right? What we do is we hook up this unfavorable chemical reaction to one that is favorable. Nature loves it when cells say, yeah, I'll break up this ATP into ADP and inorganic phosphate. That's like a dream come true for nature. It's beautiful, sure. The world's going to become more disordered, right? What's not to like from nature's perspective about releasing that energy from ATP and making ADP, right? So nature loves that. Nature hates this. Let's say to nature, here's the thing, nature, I really like to have some of these things floating around, right? So here's what I'm hoping we can do. Let's do this deal. You let me make one of these, and in exchange, I'm going to bust up one of these for you. Is that fair? Right? What do you think? This has an energy associated with it of negative 7.3 kcals per mole. Nature's 7.3 kcals per mole, beautiful, right? This has a delta G of plus 3.4 kcals per mole. Nature's bad. No, I'm never going to do that. But if you say to nature, here's the thing I'd like to do, I'll give you the 7.3 kcals per mole if you let me get 3.4 out of it to do some work. And nature's, okay, fine, right? On balance, it's a good deal. We're linking an unfavorable chemical reaction from the energetics perspective to a favorable one from the energetics perspective, and we end up with something that has a net negative delta G. So 7.3 kcals per mole minus 3.4 kcals per mole gives you 3.9 kcals per mole, negative 3.9 kcals per mole. That's how the magic happens. Again, metabolism. It's the sum total of all the chemical reactions taking place inside of a cell, right? That's the deal. There's a baseline noise of chemical reactions that are taking place in living things and non-living things. That is part of metabolism. But that particular chemical reaction, making glutamine from glutamic acid and ammonia, that's not one of the things that's taking place just by itself. It only takes place because living things facilitate it. They link that unfavorable chemical reaction to one that is energetically favorable, and at the end of the process, nature's happy because entropy has increased, and we're happy because we've got the reaction that we think is going to help us also taking place. So that's how the math of the thermodynamics aspects of this all works out. And that brings me then to what is it that is the broker? Who's the negotiator that's brokering these deals, that hooks up the hydrolysis of ATP to ADP to the synthesis of glutamine? How does that play out? The short answer, and again, I already told you this joke, right? It's a cliche amongst biologists. How does that happen? The short answer, a protein does it. right? That's a running joke. You guys haven't been, I haven't said it to you enough times yet for you to say, oh, grown, yeah, sure, a protein does it. But that's a fairly universal answer. How does this happen? A protein does it. How does that particular chemical reaction we've just been talking about? A protein does it, all right? The proteins that we're talking about here are also what we call enzymes. So enzymes are biological catalysts. Right? That's a formal definition of an enzyme. It's a biological catalyst. Let me parse that for you just a little bit more. A catalyst is something that increases the rate of a chemical reaction but doesn't get consumed or changed in the process. Right? Pretty cool concept. It'd be neat if such things existed. It turns out they do. Right? This is something that can cause a chemical reaction to take place faster than it would have otherwise, and yet that something doesn't change in the process. So you can use it over and over and over again and have that chemical reaction take place faster many, many times. It's not just a one-off kind of deal. So enzymes are biological catalysts. There are things other than proteins that are enzymes. So biological catalyst, enzyme, is a broad tent, big tent. Most of the things under the tent are proteins. And so for the rest of this class, and probably for the next couple of classes that you might take in a biology set of courses, it's fair to equate proteins with enzymes, or better, enzymes with proteins. Okay? But I want you to have in the back of your head also this idea, there are other things that might catalyze chemical reactions that living things make as well. And I'll mention one to you in particular because it's kind of important further down the line. RNA turns out to also have the ability to have some catalytic activity. So RNA could be a catalyst, an enzyme. But again, for our purposes, let's just focus on what it is that proteins do, because that makes up the vast majority of enzymes. So most enzymes are proteins. They're biological catalysts. And guess what? In the course of this class, I've given you, like in 2 lectures, all of freshman chemistry. In a couple lectures, last lecture I told you pretty much everything that's important about thermodynamics. There's a discipline, there's a series of classes that you take to learn all about enzymes, okay? Those courses, that topic is called enzyme kinetics. And if you have any vague idea that you might want to become a biochemist someday, it turns out, right? I can make fun of that class of people. I'm one of the ones who suffered through the process to be able to say I'm a biochemist. Enzyme kinetics is pretty much at the heart, it's the cornerstone of all biochemistry. Biochemists are trained to be able to pick apart a protein, say what it does, say what shape it has, say what reactions it catalyzes, to beat it up in a way so that you can figure out what molecules help it catalyze the reaction, to beat it up in a way that lets you figure out what molecules prevent the chemical reaction from occurring, right? That's what biochemists do, and that's all enzyme kinetics. All right, so you guys can look at ease now, right? There's no more pressure on you to look good for your parents later. But I was playing a game with a bunch of you before the class started today, so I suffered through a PhD in biochemistry. I still have the scars from that to this day. And one of the courses, a whole year long, was built around this book. The game I was playing is like, pick a page, any random page in this book is full of the gnarliest, nastiest, scariest mathematical equations you have probably ever encountered. Yeah, how about that? It turns out every page in this book, it's an easy bet to win. It's just nasty math. And this whole book is full of those equations. I just want to use this. I use this frequently when people come and they say, gee, you seem like you've got a pretty gig going. How can I get a PhD in biochemistry so I can be like you? And I say, this is what you got to do. This is your union card if you want to become a biochemist. You got to have some ability to navigate through a book like that. I got good news for you. There's lots to know about enzyme kinetics. Once again, there's not that much that's really important that we need to focus on for a class like this. So today, I'll tell you the best parts, the juiciest bits, the most interesting parts of everything that comes out of a book like this. Enzymes and enzyme kinetics. There's some jargon that goes with the territory when you're talking about enzyme kinetics. Most of the chemical reactions that we'll be talking about in this class, starting on Tuesday, we're going to focus on a subset of metabolism called glycolysis. We'll focus on how it is you get energy out of glucose so that you can make ATPs. But most of the chemistry that we'll be talking about for the rest of the class falls into this umbrella, under the umbrella of redox reactions. And what's happening is something is being oxidized and something is being reduced. And when you talk about a redox reaction, we need to talk about one of the things being a reducing agent, the other thing being an oxidizing agent. Notice how for the reactants here on the left side of this equation, the chemical structures that are involved, all of those electrons are high energy electrons. That's code for the electrons are shared equally between the atoms involved in the covalent bonds. Even better, and they're trying to help you graphically here, those electrons are kind of like equidistant from the atomic nuclei. They're high up, they have high levels of energy, they're high altitude electrons. So that's what's true for the reactants in the redox reaction. When those things become with products, what happens is we have an energetically favorable process, because now, instead of high energy electrons like these, check it out, those electrons are all hanging out close to the oxygen, those electrons are hanging out close to this oxygen, those electrons are close to this oxygen, low energy electrons versus high energy electrons. All right, so this is the heart of what's going on with a redox reaction. Interestingly, perhaps to some, redox reactions, you don't necessarily have to make or break bonds, but what you have to do is change the energy of the electrons. If you go from high energy electrons to low energy electrons, that is a redox reaction. Again, what we're going to be talking about for the rest of this class, in a nutshell, are redox reactions. And for any chemical reaction, redox reactions or others, this is an energy profile of how the reaction takes place. One more time. The reaction can't take place unless it's energetically favorable. You start out with a high level of Gibbs free energy, and you end with a low level of Gibbs free energy. Delta G is negative. Okay? And that's what you're seeing over here with this particular diagram. You've got reactants that start out with a moderately high level of free energy, and you end up with products that have a lower level of free energy. You release energy in the process, the delta G is less than 0. It's negative. Okay? In class on Tuesday, I told you, glucose, converting glucose and converting glucose and oxygen to carbon dioxide and water has a ridiculously good delta G, negative 686 kcals per mole. And I asked you, what's up with that? If that's such an energy-rich molecule, why doesn't it all just fall apart? We have oxygen all around us. Why doesn't that glucose just combust? Spontaneous combustion, release all that energy, poof, one big fell swoop. Why doesn't that happen? All right? It doesn't happen because in order to release that energy, to go from the reactants to the products, in order to do that, you also have to juice the system up a little bit. That's a nice colloquial way to describe it. You have to kind of get these guys razzed up a little. You got to get them excited to what's called a transition state. You can see what they've tried to do here is show you. It's a wonderful illustration, right? It shows you who wouldn't look at that squiggle and say, oh, something's happened there to really shake that bond up, right? I don't know. I guess that's the best they could do with a black and white line, right? A black line on a white background. Those squiggles are supposed to make you think, oh, this is really unstable now. It could fall apart at any second. The point is that somehow or other, we need to get these molecules excited so that they achieve what's called a transition state. And then once they're at the transition state and things are all sort of, shook up here, then you can go back either to the reactant state, or from this position, you could fall this way, or you could fall this way. If you fall this way, you release a big chunk of energy, and we've done something like burn glucose and made carbon dioxide and water. Okay? But to get from the stable reactants to the stable products, again, you got to pass through that transition state. If you do, you get a big payoff of energy. If you don't, you just hang out there indefinitely. The energy that you need to invest into this system to achieve the transition state, to get past that hump, that energy's got a fancy name. We call that the activation energy for that particular chemical reaction. It's the activation energy. A lot of times, activation energy, at least in non-living things, that activation energy just comes from heat. If you heat the system up enough, you invest enough energy into the system that you can cause the reactants to get those unstable bonds and jump up to the activation, the transition state, an excited state that allows them to fall down further to the products. Right? So that's often the way that it takes place in non-living systems. But right now, appreciate that the activation energy is a hurdle. Right? You have to clear this hurdle in order to make it through to the products and release the energy at the end of this whole process. I want to make this in bold letters for you. Big news. You know what enzymes do? You know what proteins do? The only game that enzymes play pertains to activation energies. That's it. The only thing enzymes do to cause certain chemical reactions to take place inside of our cells that wouldn't have taken place otherwise is they play with the activation energy for those reactions. What do you think? Do they increase the activation energy, or do they decrease the activation energy? Increasing it's not a good choice. I don't know why you would think you might increase it. I hope that's not where anybody's been going. Proteins, enzyme, biological catalysts do their thing by reducing the amount of energy that it takes to get reactants to the transition state so that they can become products. Reducing the activation energy results in an increase in the speed of a chemical reaction. Here's pictures that tell you the same thing that I've just been talking about. In the absence of a catalyst, the black line shows you how much energy you have to invest to get to that transition state. Very simply, you put an enzyme in there, you put a biological catalyst into the mix, and the amount of energy that you need to invest is less. It's easier to pass through that transition state. It has no effect at all. This is an important point that a lot of people seem to miss the first time through. It has no effect at all. An enzyme has no effect at all on the delta G of the reaction, right? Look at that picture there. The red line and the black line, they start at the same point and they end at the same point. Catalysts, enzymes, any catalyst cannot change the delta G. There's a certain amount of energy that's inherent in the substrates. There's a certain amount of energy that's inherent in the products. Right? To go from one to the other, for it to be a spontaneous reaction, it must be a negative delta G, but that delta G does not change in the presence of a catalyst. The only thing that changes is the amount of energy you need to use to juice the system up to get to that transition state. That's what enzymes are all about. So, you guys don't look impressed. Right? Let me give you some numbers. That's bound to win you over, right? Math, that's what you guys came to hear today. Let's talk about some practical chemical reactions. There's this one reaction that takes place inside of our bodies. Lots. We have a lot of this reaction going on. It's catalyzed by an enzyme called carbonic anhydrase. What carbonic anhydrase does, it's an enzyme, it's a protein, What it does is it takes carbon dioxide gas and it smushes it together with water to make carbonic acid. Okay? Carbonic acid's cool. It's good for us to be able to do this reaction because it's hard to carry carbon dioxide in our blood. It's easy to carry carbonic acid in our blood. And so you make all sorts of carbon dioxide in the tips of your fingers and toes, and you don't want it to hang out there. You can't move it away if it's in a gaseous form, so you make it into this thing that's easily dissolved in water, carbonic acid, and you take it from the tips of your fingers and toes to your lungs, where you reverse the reaction and allow carbon dioxide gas to go out your lungs. It's a good system. It works really well for us, but this has to happen lots. The energy associated with combining carbon dioxide and water and converting it, those are the substrates, the reactants. The product is carbonic acid. The energy associated with that, the delta G, is favorable. I think I tell you how favorable it is somewhere in the lecture notes. Maybe not. But let me just tell you, it's favorable. Negative delta G. Okay? But here's the thing. The activation energy for that reaction to take the carbon dioxide and the water and get them to that excited transition state so that they can go down the other side and become the products, the energy that you need to invest to clear that hurdle, 50 kcals per mole. Okay? You guys are getting some sense of scale here, right? Glucose, 686 kcals per mole. I don't care if you don't memorize that number, but I hope you appreciate 686. It's a kind of big number. It's the biggest number we've talked about in here so far in the context of delta Gs. ATP, 7.3 kcals per mole. That's a smaller number. 50, that's kind of in the middle, right? That's a good chunk of change. That's not something that's going to fall off the back of the truck. That's a pretty good amount of energy. That's how much energy you need to invest to clear the hurdle in an uncatalyzed reaction. But if you put carbonic anhydrase into the mix, that catalyst, it changes the activation energy for that reaction. And I don't think I'm going to insult you if I ask you, does it increase it or lower it? It doesn't increase it. Enzymes are all about lowering activation energy. If you put carbonic anhydrase into the mix, that activation energy to get to that transition state goes from 50 kcals per mole down to 0.5 kcals per mole. It's a 100-fold reduction in the activation energy for that particular chemical reaction. So this picture doesn't do it justice. If this is 50 kcals per mole, 0.5 kcals per mole is actually, the red line should be somewhere right around here. Everybody watched the Olympics a couple months ago, right? Remember, in the hurdles and things like that? You know, if I was a hurdler, what kind of hurdle do I want to use when I'm running in the Olympics? Do I want to use the 50 kcal per mole hurdle, or do I want to use the 0.5 kcal per mole hurdle? I mean, come on, seriously. That makes a big difference in terms of how easy it is to clear that hurdle, in terms of how easy it is to get that chemical reaction to take place. I can see people are imagining me trying to jump over hurdles. It's humorous. Go ahead, let it out, and then we'll get, okay. So what does this translate to in practical terms, right? I think now very cerebrally, very intellectually, you can say, oh yes, I understand. By reducing the activation energy, I can make it easier for a chemical reaction to take place. Good. That's what enzymes do. That's what biological catalysts and catalysis is all about. But practically speaking, you know what that does? Is that causes that particular reaction to go fast, to go much faster than it would have in the absence of the catalyst, right? Just as I'm hurdling over my half-inch tall hurdle as opposed to my 50-foot tall hurdle, or 50-inch tall, 500-inch tall turtle, well, you know, you can go fast. You know how fast, if you've looked at the lecture notes, you do, you know how fast that reaction takes place in the absence of a catalyst? We've got quintillions of carbon dioxide and water molecules in our bodies right now, right? A ridiculous number of those. It's not happening at you'd have to go a day or so before even one of those carbon dioxide molecules and a water molecule just by chance hit each other in just the right way with just the right amount of energy to clear that transition state and get through to the other side. It's not going to happen in your bodies unless you put a catalyst in there, unless carbonic anhydrase is there to reduce that activation energy. The reaction is energetically favorable. It would have occurred. It's so darn hard to get the situation just right to clear the hurdle in the absence of carbonic anhydrase. When carbonic anhydrase is present, there's been a big buildup, right? This had better be an impressive number, right? When carbonic anhydrase is present, the reactivation energy is lower. Sure, got it. It's a catalyst. That's what they do. And that makes the chemical reaction take place faster. And faster is an understatement. Faster here means 600,000 reactions per enzyme per second. Every carbonic anhydrase enzyme converts 600,000 carbon dioxide and water molecules into carbonic acid every second. Again, drop the mic, walk away, I'm done. I can't do much better than that for you with this material, people. I'm sorry. Come back later when we're talking about DNA and genetics, and I've got better stuff for you to work with. But oh my gosh, that's what this game does. That reaction wasn't going to take place unless we helped it along with a catalyst that we've introduced to the system to help it along, right? So metabolism, it's the sum total of all the chemical reactions. Sure, carbon dioxide and water to carbonic acid, that's going to be happening at the background and baseline sort of stuff, but it is so infrequent that you could just more or less forget about it ever taking place unless you help it along. That's what living things are doing, is they've gamed the system, they lower the activation energies, and they let the reactions that they need or that they want to take place at a faster rate than they would have otherwise. Carbonic anhydrase is a speed demon. 600,000 reactions per second, that's at the high end. There are others that are faster. The lecture notes give you another example of one that's 40 million reactions per second, so that gives you a sense of scale again. But typical enzymes have turnover rates. That's the speed number that I'm talking about, turning over substrate to product. Typical enzymes have turnover rates anywhere from about 100 to 1,000 conversions per second. All right? Still crazy fast. I mean, you can't type that fast. You can't type 100 letters in a minute. Well, maybe. But 1,000 in a minute's getting kind of hard. We're not talking about minutes. We're talking about seconds. We're talking about molecules going from substrates to products. That's why the reactions taking place in living systems are different than the ones taking place in non-living systems. It's because we're given a pass in regard to activation energy to the ones that we like. Okay? So if a reaction takes place inside of a living thing, there's a pretty good chance it's been catalyzed by an enzyme. And if there's an enzyme there, that probably means it's working to the advantage of the cell, the living thing, to do that. Otherwise, wouldn't have put the enzyme there to catalyze the reaction in the first place. So I've been talking about enzymes for a while now today, and I've flashed a picture of an enzyme to you early on when I was talking about how proteins fold up into secondary and tertiary structures and all that stuff. Here's a picture of an enzyme for you. I remember telling you, when you look at the three-dimensional structure of an enzyme, the active site where the catalysis takes place usually hits you in the face. It's usually pretty obvious. I don't know, maybe it's because I've seen these more times than you have, but come on, seriously, would it be hard for you if I scrubbed off the active site and the substrate? You'd probably figure, there's something happening where that active site is. It's kind of conspicuous that there's something going on. In fact, you may even start to think, I find myself thinking, that all the rest of this stuff is really just there to help keep this stuff in a particular conformation. One more time, in living things, structure and function one and the same. They're intimately related. The shape at that active site, the charges of the R groups, the hydrophobicity sometimes of the R groups that are brought to bear right there, that's what determines the function of this enzyme. And you can see a substrate, this little red thing, might fit comfortably into that shape. Check it out. And this is actually true to life. This is not like a cartoon. Check it out. When this substrate hooks up with this enzyme, there's a mutual attraction that's going on here. When the substrate comes into the active site, not only does it find a nice, comfortable spot to sit, but the rest of the enzyme gets kind of excited too. And it says, oh, look, the substrate, now I'm whole. I can come back and, oh, look at this. It bends over and it wraps around and it nestles that substrate. It's not just the immediate points of contact. Once the substrate's in the active site, the enzyme typically changes its shape in a way so that it has even more intimate contact with the substrate, like what you're seeing there in that B part of the picture, the enzyme substrate complex. So how does this work to lower activation energies? Here's how this works to lower activation energies. Because if enzymes can't lower activation energies, they're not good for anything, right? We don't talk about enzymes unless they can lower something's activation energy. Because if they don't lower an activation energy, they're not a catalyst. And if they're not a catalyst, they're just taking up space. Maybe even worse, they're using amino acids that we could have used to do something that actually did get the job done. Okay? So how does an enzyme pull this off? How do you reduce an activation energy? Here's how it plays out. You get your enzyme. Now this is kind of cartoony, right? But imagine an enzyme that looks sort of like this, and it's got a really obvious active site. This is like a caricature of an enzyme, right? Big honking active site. Look at that. What if we had something that poked up a little bit there? That might be a good substrate. What if we had something that's kind of pointy and poked in there? That might be a good substrate. That sort of thing. It's a cartoon. Check it out. Here are some substrates for this cartoonish enzyme. One of these substrates fits neatly into one spot of the active site. The other fits neatly into the other part of the active site. And hey, look at that. The two substrates kind of mesh together pretty well when they're all there at the same time. This is the enzyme substrate complex in a more cartoony version than what you saw on the previous slide. This is going to reduce activation energy. Those two substrates, the red thing and the green thing, for those of you who are colorblind, the thing on the top that's got the rounded pointy thing and the thing on the bottom with the triangle pointed thing, those two substrates could have smushed together of their own, right? You don't need an enzyme. They have a flat thing there, right? Those two flat surfaces could have come together, right? If they came together, if those two molecules, the red thing and the green thing, hit each other at just the right angle, with just the right speed, temperature is what we're talking about. If they came together in just the right way, bam, you make a product. Everything's cool. The reaction takes place. If this is an energetically favorable reaction, you can have that happen without the enzyme there. But check out what goes on when the enzyme's there. Let's say the red molecule shows up on the scene first. Okay? It can stick to the enzyme at the active site and get held there. You don't have to have the red thing and the green thing show up at exactly the same instant. The red thing can get there whenever the red thing gets there, and it'll be held there by the enzyme. And the green thing can get there whenever the green thing gets there. Maybe the green thing gets there first. Who knows? But once it gets there, it gets held there by the enzyme until the other thing comes, and then they're right next to each other. Time gets taken out of the equation. You don't have to have the timing exactly right to get those two things together. The active site holds the substrate there until the other substrate shows up. Time isn't important. That alone should increase the rate at which these reactions take place. And check it out. Apparently, when the red thing and the green thing come together in this cartoon, those substrates can give rise to products, a blue thing and a yellowish thing, right? They can swap some bonds. And once the products are formed, the enzyme can release the products and come right back like a good old catalyst, because remember, catalysts are neither altered nor consumed during the course of the reactions they're catalyzing. The enzyme comes back and it's ready to do the process all over again. If that purplish enzyme, and for some reason enzymes are almost always shown in purple, it's not just in your textbook, it seems to be a convention amongst illustrators for textbooks. But if that enzyme happened to be carbonic anhydrase, that cycle happens 600,000 times a second. And the red thing and the green thing are carbon dioxide and water, and the blue thing and the yellow thing are one thing called carbonic acid. They don't actually form two molecules, it's just one molecule that comes out. Maybe carbonic acid with a hydrogen floating around, if you really want there to be two things. But check it out. That's what's going on here. That's how the activation energy is being reduced. That's how it is that the enzyme is facilitating this particular chemical reaction. In some sense, it's as simple as that. Structure and function. So important in biological systems. The structure of an enzyme has a lot to say about what the enzyme does and how well the enzyme does what it does. You guys know from your own experience in the lab, a week ago? 2 weeks ago? No, 2 weeks ago maybe. You guys know from your own experience in the lab that enzymes actually work better at some temperatures than others, right? So check it out. There's an enzyme, doesn't say what the name of the enzyme is, but this particular enzyme has optimum rate of activity. And I'm just going to use the technical, I have the right to use it. I've studied this book. Turnover, right? The turnover rate for an enzyme, this enzyme is optimal. You have the highest turnover rate at body temperature, 37 degrees. That doesn't mean the enzyme doesn't work when you're cold and dead, right? At 20 degrees, it just doesn't work nearly as well. It works at about half the speed that it does when it's at its optimum temperature. Guess what? Over the course of evolution, those enzymes have evolved, they've been tinkered with, they've been tweaked, so that the combination of amino acids and the structures they have adopt to just the right conformation at that 37-degree temperature. I'm pretty confident that if our body temperatures were pegged at 35 instead of 37, the optimum temperature for all of our enzymes would have been lower because there'd be different, slightly different conformations of those proteins because of subtle differences in their amino acid sequences. And in fact, if you look at something that lives at 77 degrees centigrade, deep sea thermal vents where the water's almost boiling, really hot, those enzymes work optimally at 77 degrees. Shouldn't surprise you to hear, I hope, that if you change the pH of an environment, you can change the structure, the shape of a protein, the functionality of a protein. Some enzymes, like pepsin, work best at very low pHs. That's the conformation where they work best. That doesn't mean that they don't work at higher pHs. It just means it works there. Others work better at really high pHs and don't work very well at all at lower pHs. So temperature and pH shouldn't surprise you to hear. Those things can influence the turnover rate for an enzyme because they'll change the structure and therefore the functionality of the enzyme. But that then brings me to talk with you about this idea of inhibitors and activators, and also to introduce you to a word that I'm going to use a lot and you may be able to use to impress people with, right? Let's get that out of the way first. Allosterism. Allosterism is what I'm going to be using to tell you the shape of something has changed. Okay? That's all that it means. But in light of all the things I've just been talking about, you change the shape of an enzyme, You're going to change its functionality. You're going to change its ability to catalyze a reaction. So an allosteric effect, or allosterism, that means we're talking about a change in shape that results in some change in function. And I'm deliberately being kind of ambiguous here. I'm not saying that it increases the functionality or it decreases. It could be either. Anything that changes the shape of an enzyme in a way that causes the functionality of the enzyme to change, that's an allosteric effect. Some allosteric effects increase the functionality, some decrease. Here's the thing. If they increase the functionality, we call that an activator. If they decrease the functionality, what would be a good one? An inhibitor. How about that? Let's go with that one. All right? So allosteric effects, allosterism, we're talking here about changing the shape of a molecule, of an enzyme particularly, in a way that causes its functionality to be changed. It's all about control, right? For living things, we want to control. We want certain chemical reactions. Want's not the right word. We need, it's necessary to keep the other stuff running. We need certain chemical reactions to take place. And we play a game. We facilitate this by creating and deploying enzymes that help us accomplish that goal, to get certain chemical reactions to take place. But stop to think for a moment. Times change, circumstances change. Sometimes the right chemical reaction to take place 10 seconds ago isn't the right chemical reaction to take place right now, right? It wouldn't be a particularly good, well-designed system if once you made an enzyme, it just stayed there forever, right? Wouldn't it be swell? Wouldn't you be able to exert more control over the chemical reactions taking place inside of your cells if once you've made the enzyme, you could turn it off? That's a neat design feature. You know, having light bulbs is swell, but it's also nice to once in a while be able to turn those light bulbs off, right? Wouldn't it be swell if there was a switch, if there was something we could do to turn an enzyme or a light bulb off? And by the same token, once you've turned the enzyme off, maybe what you really want is to have a quick response. Maybe you want the enzyme hanging out there ready to go but not active until you tell it to kick into gear. Now's your time. Do your thing, buddy. This is your time. It'd be nice if sometimes we could turn them off with inhibitors, and it'd be nice if sometimes we could turn enzymes on with activators. Might be nice if some enzymes were responsive to both inhibitors and activators, but pretty much every enzyme has to be responsive either to an inhibitor or an activator in order to maintain control and to have responsiveness, to be able to respond quickly as you might be able to a change in circumstances for a cell. And again, that's ultimately what inhibitors and activators are going to all be about. So let's talk here about we'll start on the positive aspect of it. We'll talk about an activator. Check out this enzyme. This enzyme I would call as a trained biochemist, right? Once again, I speak from authority here. As a trained biochemist, I would tell you, this is a tetramer, right? It's a protein, it's a set of 4 proteins, 4 copies of a single protein that have to come together, quaternary structure, right? 4 proteins, 4 copies of a protein need to come together in order to be able to catalyze a particular chemical reaction. Apparently what needs to happen is a substrate needs to fit into this very obvious active site, right? But these monomers in the tetramer, maybe the active site, maybe this thing here is kind of floppy and sometimes falls in on itself. An activator would be a molecule that can interact with this enzyme in a way that locks the active sites into their open active form. Because the green thing is here, this little thing can't flop over and cover the active site. And because the green thing is here, this thing can't flop over and cover that active site. That's all that's going on here. Nothing more complicated than that. How does the green thing do it? Well, there are probably certain R groups on this surface of the monomer and certain R groups on this surface of the monomer that really like to nestle up against whatever it is that's on the surface of the green thing, right? Where there's a negative charge on the green thing, there's a positive charge on the protein. Where there's a negative charge on the protein, there's a positive charge on the green thing, right? Opposites attract. But that green thing locks that enzyme into an active state, ready to go. Whenever a substrate shows up, I got a spot for you, big boy. You sit down right here, and I'm going to hook you up. A little while later, something else is going to come along, and beautiful things will happen, and we'll catalyze the reaction. Life's going to be good. And that's in contrast to what's going on with this red thing. The red thing seems to like to target the same spot on the surface of this protein, but when the red thing is there, it causes the behavior of the enzyme to change. It causes the active site to be masked. That floppy thing comes over, covers the active site. No room at the end. Sorry, you might be the substrate that I usually like to play with, but sorry, today we're closed. No catalysis happening here. So, activators are things that make it easier for substrates to get to the active site. Inhibitors are things that make it harder for things to get to the active site. If substrates can't get to the active site, that's going to have a negative effect on the turnover rate for the enzyme. Obviously, I would think. Check this out. Here we have an instance of cooperativity. This enzyme doesn't have active sites that are very open for business, but if one substrate manages to fight its way through, getting that substrate in contact with the enzyme causes the rest of the binding sites to also open up for business. Hemoglobin, the thing that carries oxygen in our blood, that's how hemoglobin works. It's usually not that receptive to binding oxygen, but if you get one oxygen molecule to bind, the other 3 sites for oxygen binding become much better at taking on oxygen. It turns out that that's a neat design feature in a sense because it lets you pick up oxygen where the concentrations of oxygen are high, and it lets you deliver that oxygen, release it to places where oxygen concentrations are low, because it also works out that when one comes out, the other 3 are easier to kick out the other 3 oxygen molecules. So, inhibitors and activators, that's how they work. Probably didn't need me to tell you that. Could have figured that out on your own. Well, let's have some rubber hit the road here. How are we going to get these enzymes to do something useful for us? You got to hook up unfavorable chemical reactions to ones that are favorable from a thermodynamics perspective. You saw that with the synthesis of glutamine, one of the 20 amino acids, right? We link that to a hydrolysis of ATP, right? So that's one thing that's in play here. Another thing that's in play here is you can have inhibitors and activators that kind of let you, once you've made the enzyme, you're not committed to having the enzyme do its thing. You can turn it on, you can turn it off. Depending, as the circumstances change, you may decide a cell may find its needs change. And there are ways to call the horses back to the barn after the door's been open. Right? Is that the right expression, or is it cows that leave the barn? I don't know. Right? But nonetheless, right? So you there's an energetics thing you need to take into account here. There's a reduction of activation energy in terms of speeding up reactions. That's another kind of big point so far today. And then there's this business of having the reactions take place more quickly and sometimes causing them not to take place at all. But the slide on the screen right now is showing you a real practical application of a lot of the stuff that I've been talking with you about in fairly abstract terms so far today. So we talked earlier about making glutamine. Well, another amino acid you need right now is isoleucine. Can't make your proteins without the amino acid isoleucine. Where does isoleucine come from? It doesn't grow on trees. Actually, I guess it does grow on trees, but for our purposes, where's the isoleucine coming from? Did you know, would you have guessed, that it starts off as something called threonine, another amino acid, right? Would you have, I would have known. Well, what a happy bit of insight we have. But it's not trivial to go from threonine to isoleucine. It's actually a what? 1, 2, 3, 4-step process, maybe 5. It's a 5-step process to go from threonine to isoleucine. How do you get those steps to take place? A protein does it, right? Not just, each of those steps gets its own protein. How do you accomplish that first step in the process to get to intermediate A? Oh, a protein does it. Right? That's the cliche answer. How do you get from B to C? A protein does it. Here we can be even a little more specific. We can say an enzyme does it, a special kind of protein, one that catalyzes a chemical reaction. I didn't say it earlier, but boy, I would be doing you a huge disservice if I didn't make it really clear that this enzyme is really very specific for that substrate, okay? This enzyme is really good at interacting with threonine and not at all good with interacting with anything else. And the reason for that is the active site is really very specific. There's a certain combination, it's got a certain shape, a certain combination of charges and hydrophobicities and hydrophilicities. Only this substrate can fit comfortably into that site. Molecules that are very similar to this might come in, but they won't fit in as neatly and as quickly and as stably as this particular one does. So this enzyme is all about converting threonine to the first intermediate on the way to making isoleucine. And the product that gets released from this enzyme fits perfectly into the active site of enzyme 2. And enzyme 2 is all about converting intermediate A to intermediate B, and it really does nothing else. And the same for 3 and 4 and 5. We have a set of 5 enzymes, each of which has a very specific, very specialized task, catalyzing a chemical reaction that lets you ultimately convert threonine to isoleucine. Right? So check this out. What if you've got plenty enough isoleucine? Thank you very much. I don't need any more isoleucine today, thank you. Right? What do? One of the things you should probably do, and I'm talking to each of you in your cells, what you should be having your cells do, if you've already got all the isoleucine you need, stop converting threonine to isoleucine. Right? Don't do it anymore. You already got isoleucine. Stop it. Cut it out. Why wouldn't you want to do that? Threonine's kind of useful in its own right. You might have things that you need to use threonine for, like making some other proteins. Right? So if you already got isoleucine, stop chewing up all your threonine. Use the threonine. Don't make it isoleucine. That's one reason to not make isoleucine if you've already got isoleucine. Another reason is each one of those steps has got to have a negative delta G associated with it. You're burning energy. Burning energy is a bad idea. Don't be wasting it. Threonine is swell. Keep your threonine. Later, when you need isoleucine, then we can talk. Right? So there's a number of reasons why you might not want to convert threonine to isoleucine if you've already got enough isoleucine. So how are you going to pull that off? Right? Noble ambition. Obvious that you shouldn't be doing this whole chain of reactions if you already have isoleucine. How are you going to pull it off? The picture tells you. You've already figured it out, I'm sure. If the picture wasn't there, we could take this through a little bit more Socratically and sort of stepwise. But part of the answer should probably involve an inhibitor, right? And if part of the answer is an inhibitor, what's that mean? That means that we're going to have a molecule that comes back and tickles one or more of these enzymes and tells the enzymes, stop, don't do this anymore, right? So there should be an inhibitor involved. When you're low on isoleucine, it sure would be nice if you could get that inhibitor out of the way so that you could start the process over again really fast. You don't have to wait for the enzymes to get made. They're sitting there ready to go. That would be nice, right? So when isoleucine levels get low, well, okay, turn it on now. I want isoleucine now. I don't want to wait 5-10 minutes for you to make the enzymes. I want isoleucine now, right? So turn those enzymes on. So we need an inhibitor. We need that inhibitor to be tied into isoleucine concentrations in some way, right? Conceptually, how would you design this? If you were God and you were tasked with coming up with this pathway, sit down with a piece of paper and a pencil, what would the system be? I'm going to have a chain of enzymes catalyze the reaction. I guess I have to have them be negative delta Gs. Okay, I'm good with that. I want it to be sensitive to isoleucine levels. What could I use that might be directly related to isoleucine concentrations in a cell that I could use as an inhibitor? Hey, oh, what about this? How about if I use isoleucine? What do you think? Brilliant. What a swell idea. Here's the thing. If I've got a lot of isoleucine already, I want isoleucine itself to be the inhibitor that stops the synthesis of more isoleucine by the enzymes that are involved. And if you figured that out, wearing your God hat, this next thing is trivial, right? That one's that maybe you have to think a little bit to come up with the fact that you use isoleucine as an inhibitor. Where do you want to do the inhibition? Do you want the inhibition to be at the end of the process, in the middle of the process, at the beginning of the process, all through the process? Nothing wrong with all through the process, but that's kind of tricky. It's hard to pull that off. You know what's real easy? Shut it off at the start. Have the inhibitor of this metabolic pathway shut down the activity of the very first enzyme in the process. Again, simple, but brilliant. That's exactly the way, if you were being a clever engineer, you would design this system, isn't it? What's going on the screen here? Oh my gosh, that's how it is that this pathway gets regulated. This metabolic pathway is keyed in to the isoleucine levels. When isoleucine levels are high, you know what happens? Isoleucine itself sits down on the first enzyme in this metabolic pathway, has an allosteric effect on that enzyme, changes the shape of the enzyme in a way that makes the active site no longer receptive to threonine. If threonine can't bind to the active site, it's over. None of the rest of this happens because you're never going to make intermediate A to tickle enzyme 2. You're never going to make intermediate B to tickle enzyme 3. You shut the whole thing down. I suppose you could have had isoleucine stop the activity of this enzyme too, but what's the point, really? You've wasted a lot of threonine by the time you got to this point. Really, the best place is to just close the barn door before you ever burned your first threonine and stop that process from taking place. Nip it in the bud. Beautiful, right? Eminently logical. And the student evaluations at the end of the class, I pore over those. I look at them really carefully. I often will see students say, that Crane guy makes really simple things seem complicated, right? This is a really simple thing. You didn't need me to tell you this. You just needed to sit down and think about it for a little while. What's the most logical? What's the best way to go about accomplishing this? You're going to find time and again in a class like this, the way that living things have worked it out is exactly the way that you would have worked it out if you were thinking about it and trying to come up with the most efficient, logical process that you could. It's beautiful. It makes the hairs on the back of your neck stand up, right? This is neat stuff. So we got a fancy word for this kind of process. This is feedback inhibition. Really makes a lot of sense, right? The product is what's acting as the inhibitor. Notice that the site that the inhibitor is binding to is different than the site that the substrate binds to. It's not on the slide, the words I'm going to give you, but this is called non-competitive inhibition. And that's in contrast with competitive inhibition. Right? So there's typically, we usually parse inhibitors, we put them into two buckets. We put those that bind to someplace other than the active site into a category we call a non-competitive inhibitor, and we put the ones that bind to the active site into the category we call competitive inhibitors, right? That sort of explains to you how it is that inhibitors can work. They sometimes can have an allosteric effect that closes off the active site. Sometimes they just physically compete for the same real estate on the surface of the protein that the substrate binds to, right? Those are competitive versus non-competitive inhibitors. Notice that all of this kind of inhibition we've been talking about is reversible. If isoleucine levels get low inside of the cell, you know what's going to happen? That isoleucine is going to diffuse off of its binding site, and you're going to turn the spigot on again, and you'll make more isoleucine. It's a reversible process. We haven't killed the enzyme. It's still there. And in fact, the instant that isoleucine levels get a little bit low, we can have it do its thing again. Again, we don't even have to wait for this enzyme to get made. It's sitting there ready to go as soon as isoleucine levels get low. Well, what's next? I'm at the end of the outline. The thing that's next is in class on Tuesday, we're going to focus on a set of 9 chemical reactions. With isoleucine synthesis, you saw a 5-step process. Tuesday, I'm going to talk with you about the 9-step process by which we convert glucose to pyruvate to extract some energy. That's what's in your near future. Have a great weekend. See you guys Tuesday. How does what? How does what know to turn on and off? How does the body know when you turn isoleucine off? It's all cued to